All right, this is part 5 of the cell. We are continuing our tour of the cell, identifying these subcellular structures, these organelles, and identifying their function and maybe some other information that is noteworthy about each of them. So we're moving on to the organelles. We're moving on to the endoplasmic reticulum. And really, out of all of this, I want you to know that they are made out of membrane, phospholipid bilayer. Continuous with the nuclear membrane, yeah, that's great. There are two varieties, rough ER and smooth ER. And so what we're going to do is we're going to take a look at each and identify their functions, even though in the previous part we already identified the function of the rough ER. So here's the rough ER, and then here's the smooth ER. So the rough ER has ribosomes, and it will produce proteins that get exported or incorporated into the membrane. This is just another way of saying that. Right? So that we already know that the rough ER makes proteins that get shipped out of the cell or incorporated into the plasma membrane. Smooth ER, one of the things that I want to kind of go over right here is that we're going over the major function of each of these organelles. And in the very first part, I mentioned how every single cell doesn't have all of these organelles present. Well, another thing is that the function of these organelles can change slightly depending on what particular cell we see it in. And the smooth ER really affords us a great opportunity to do that. So what I'm going to do is I'm going to give you a general function and then two tissue-specific functions. The general function will be lipid metabolism. Now be careful here. Some of you guys are going to make flashcards, which is understandable for this, and you're just going to have function of smooth ER, lipid metabolism. And then when I say some of the functions of the smooth ER could include the synthesis of cholesterol, and you're going to say false. Keep in mind, this term lipid metabolism actually encapsulates quite a bit. We've talked about all different types of lipids, and we know metabolism consists of building and breaking down reactions. So breaking down phospholipids, that's lipid metabolism. All right, so just understand that. Two tissue-specific functions in the liver. We're going to see drug detoxification. Most drugs will be detoxified by the liver, as an FYI, since this is anatomy. The other one that does quite a bit of detoxification or metabolizing, getting rid of it, will be the kidneys. So many times, if there are pharmaceutical interventions going on, depending on what the drug is, they might have to test for liver enzymes or kidney functions, and then kind of alternate the medicine, what drugs they're using, depending on how the liver or the kidneys are handling it. And then the last one I will show you because it's already typed up here. So you don't have to worry about these. This is just an example. You can see synthesis of steroid-based hormones, transport of fats, okay, detoxification of drugs. And then the third one is going to be in muscle, storage and release of calcium. Okay, so lipid metabolism is the general function. The liver, drug detoxification. Muscle, storage and release of calcium. All right. All right, so next we have the Golgi, just as an FYI. It can be called the Golgi complex. It can be called the Golgi apparatus. It can be called the Golgi body. Also keep in mind, in the future, we'll be talking about Golgi tendons, and that is something completely different. My apologies, I didn't come up with the nomenclature. So this Golgi, made up of membrane, that's all you need to know. for right now, made up of membrane, and its function is the modification and packaging of proteins. So if something needs to be shipped out of the cell, it's the Golgi that is going to handle it. And so the Golgi will receive these vesicles, modify these protein, and then ship it out. Right, so hopefully you can start to see some sort of connection between all of these organelles. And that's the other thing that I want to make sure that you take away from this, that you don't just memorize these organelles and their functions. I want you to be able to see the connection. So think about this. We have the rough ER. The rough ER has ribosomes attached that were made from the nucleolus. It's going to make a protein. It will pinch off because that rough ER is made up of membrane. So it'll just pinch off and make this vesicle. This vesicle will then fuse with the Golgi because the Golgi is also made up of membrane. And as the protein is as it's working its way through, it's being modified. Most of the times it's just glycosylated, sugars are being added, and then it is going to leave. And think of this as like having some sort of barcode. The barcode will dictate where the new vesicle is going to go. And we know if it's making a protein, that this will then go most of the time to that plasma membrane to either dump off its protein or to incorporate that protein into the plasma membrane, because it was ultimately a protein made by the rough ER. All right. We also have lysosomes, and there is this whole list that's going to follow. If we don't have enough calcium in our blood, our body will take that calcium from the bones. So we have these cells called osteoclasts, and those osteoclasts will start to degrade the bone, releasing the calcium into a soluble form into the blood. We have macrophages. We have white blood cells that will recognize something and eat it. And once it eats it, we want to kill whatever it just ate. And we want it to break it down into its parts. So lysosomes are the ones that will do that. So the take-home message here, by no means do you need to write and memorize all of this. The take-home message for these lysosomes, if something needs to be degraded, be it for defense, for maintenance, or for digestion, any reason, if something needs to be broken down, it is the lysosomes that will take care of it. All right, so what we see is all of these membranous structures, Golgi, rough ER, smooth ER, lysosomes, all of that, and this kind of goes back to what I was saying earlier, that I want you guys to know how all of these organelles work with each other, all right? So what we have are these organelles that are all made up of membrane that make this endomembrane system. I would worry less about what's on here and more about kind of what I was saying. I will devise some sort of test question that makes sure that you can follow, let's say, the production of a protein, follow it through to all of the organelles that are going to interact with that protein. All right. So just focus more on that and that relationship than anything specifically on this slide. All right, and that's the end of this part. There is one more part.